Right, moving on, it's my pleasure to introduce one of our collaborators, of course, the lead facilitator for having this conference in San Diego, which we're very thankful for, and for just being a resource for many families when they receive a new diagnosis or when they're really stuck on not knowing where to take the next steps. And that would be Professor Hudson Freeze, the director of the Human Genetics Program at Sanford Burnham Prebys Medical Discovery Institute. He's worked on glycobiology for over 40 years, with the last 22 years focused on the identification and understanding of human glycosylation disorders. The Hud Freeze lab has now discovered, which we just had in a prior presentation, 21 human glycosylation disorders, and at present it is the only laboratory in the United States primarily devoted to studying CDG. So, welcome, Hud.
Thanks, Andrea. We're all set there? Okay. Hey, thanks everybody for being part of this meeting. This is just tremendous to see so many people here and to have everybody stick through the scientific sessions. I really applaud your endurance for that. A lot of us in science don't stay that connected. Before I actually start in, if there are people who have sent us cell lines but have not signed your consent forms, Bobby is here. Bobby, stand up. There he is. Bobby has consent forms. If you want to sign your consent form sort of prospectively, thinking that maybe you're going to take advantage of that and send us cells, blood, or whatever it may be, again, talk to Bobby, because we have to have these things, otherwise we're sort of frozen in time. Okay, look, I'm going to try to keep this a bit on the short side so we don't run over time, and I know we're already behind. But this is sort of CDG updates and some of the perspectives that I've put together. And for this group, maybe this next question is not really appropriate.
What's the matter, Sal? Not up on your glycobiology?
That's a clip from the movie Extraordinary Measures. That's Harrison Ford talking about glycobiology. And so I usually use this to an audience of scientists because most of them are not up on glycobiology. And I think this group is probably farther along than most of them. And so if you need help in trying to understand the basics of this, write to us. Let's see if we can get some sort of a consensus of a way to get more information about basic glycobiology. So I'm going to talk about several different things within the research findings. I'm going to talk about the new diseases and the trends that we're starting to see. We'll touch on exome sequencing, and my apologies, I had to be away for some of the earlier talks, so if this is all old stuff to you, just start going, we've heard it all, and shout me down and I'll move on to something else. The importance of verification, and I'll tell you what I mean by that, and then how to verify. And then we're going to get into therapies to some extent. So if you look back over the, I guess, where are we now? The decades, it's a good way to say it. We have, there we are. So here we are now. Depending on who you talk to and how you count the disorders, we're now really up to 130 different types of glycosylation disorders. Those are things that affect glycosylation. Now, it may not be in this pathway that we talk about. It could be things that are influencing it, but in the end, it changes glycosylation and has an effect on your child. So these are many different pathways, and I don't think it's so important that you understand all that, but a couple of things I want to point out. Here are the years down here, and here is the growth in the number of disorders, and as you can see, it has just exploded. Back here in 1981, I was working in glycobiology, and I was working on the first, we didn't call it this, but it was the first glycosylation disorder. It was called eye cell disease, and this back in 1981. And there has been a lot of interest in that, and the basic scientists began to understand what was wrong only because there were patients, and those patients had deficiencies in adding a molecule called mannose-6-phosphate onto sugar chains, and those were very particular proteins that carried that. Those were ones that were going to go to an address within the cell called a lysosome. And a lysosome is a little digestive garbage can that is very acidic. And so you had to figure out a way to traffic proteins that were going to do all this degradation. You had to localize them in this thing called the lysosome. And it turned out that a slight modification on a sugar chain, the addition of mannose-6-phosphate, was the key to getting those enzymes to that location. And if you didn't have it, instead of going to the right place in the cell, these enzymes were just thrown out into the circulation. And that was a key finding for the common feature that united all of these things together. That was kind of the start of a good aspect of glycobiology. Now, I have a reference up here that is very recent. I don't know if you can read it. If you can't, write this down. PubMed ID number, PMID, you can look it up on PubMed, 27558841. That's a scholarly review. So, it's geek talk. But it's the biological roles of glycans. And they actually used this, an earlier version of this graph to show the growth of the importance of glycosylation. So here is one review article that tries to encompass everything, and you can't do it all in one review article, but one of the driving forces is the fact that we have all of these glycosylation disorders, and you all are the experts in those things. We're not. So keep it up. We'll keep it up. We'll see how far we get here. So we have talked about the importance of exome sequencing. Now, that was in fact one of the questions that was given to us for the Q&A period. And how has exome or genome sequencing, the stuff that you've heard about, the latest technologies, how has that really impacted everything? And Bobby, primarily, and with a little help from me, wrote a review article that should be coming out in the not distant future, but where we measured and we counted all of the disorders that were discovered between 2010 and 2017. And we divided them up by these different pathways which have importance to us as glycobiologists, but for you, unless your child has one of the particular pathways, may not be as meaningful. The point is that, look at this. There were a total of 33 N-linked, as we call them, disorders solved, and 25 of those, 25, were solved by exome sequencing. In a thing called glycosaminoglycans, 5 out of 8. Dystroglycanopathies, you heard from Kevin Campbell, 4 out of 7. Down here, the GPI anchor pathway, which many of you will know, that's got 12 out of the 14. So totally, out of 67 disorders, and think about that, there are people who are starting in medical school who have not yet finished their medical training when some of these are coming out, and yet that's the new stuff that they're going to be expected to learn. So you can imagine the load that goes into this. Out of 67 disorders, 51 were done by exome sequencing. So why is that important? Well, it's important because that is the technology that has enabled essentially all of the progress that we've seen in the last 7 or 8 years. And the cost is dropping. I went on the internet this morning just to see what I could find, and there are places that will talk about doing an exome for $1,000. Many of the places you can go to, commercial firms, for trying to get data on what your child may have will be about $6,000, and that's for mom, dad, and the child. Perhaps a little more if you have a sib in there. And this is cheaper than a lot of the tests you'd be charged for in going to the hospital. So the idea of doing exome sequencing fairly soon in a diagnostic odyssey is becoming the norm, not the exception. And many insurance companies, and I don't know the details, are covering this. So stay up with it. Make sure you know about those sorts of things, because there's going to be other people who will come to you as parents of CDG children, and they're going to ask you, what do we do about this? How do we know? Where do we go? You can be of help. And so that exome, the cost of exome sequencing is certainly dropping. And it's going to be something I think that is going to completely change the complexion of medicine going forward. And if you remember Dr. Hood's lecture, he talked a lot about how all of this new technology is changing the way we do everything. So let's just talk about some of the more recent developments that have taken place in the last several years. So first of all, I think, did people get to hear about the difference between de novo mutations and inherited mutations? Is everybody up on that? Raise your hand, raise your hand. So a few, not everybody. Okay. This was covered in one of the sessions. So generally what happens in most of the disorders of CDG, but not all of them, you inherit one gene from mom and one gene from dad that is defective. It's only 1 out of 4 times when those 2 copies of a bad gene come together in the child that you have a CDG. Okay? So and that's why it's so important to see that mom and dad contributed these mutations to the child. On the other hand, what we've started to see, and there are a couple disorders, namely ALG 13 and SLC35A2, which is the UDP galactose transporter. If that language means something to you, great. If it doesn't, don't worry about it. The point is that there are mutations that are popping up, sometimes at very recurrent positions, just so stochastically. That means randomly. So either in the gametes, the egg or the sperm from mom or dad, there can be mutations in some of the populations of those cells. And if one of those cells is the founder for the child, then that child can have a disorder. Mom and dad won't have it, the child has it. It was a spontaneous mutation. Another way to do this is that very early in embryonic development, a mutation occurs. And that mutation can then be propagated in many, many, but not all of the cells in the body. And so what this does is, if you only do a genetic analysis, let's say, of blood, you may actually miss it because it's not the blood that was affected. So this gets very complicated. So a lot of de novo mutations are coming up, and we're sort of tuned into that, and we're trying to understand how those things may take place. I mean, you can even imagine that there would be a de novo mutation. Maybe it's a very mild mutation in some way, but if you take that mild mutation and then you put a stress on the person, maybe that's when the situation essentially catalyzes some aspect of a disease. So not only do you have the mutation, you need an extra punch to do something that actually begins to reveal the consequences of it. So it's convenient for us that there are now some CDGs that are starting, because they've been known to be associated with different phenotypes, such as epilepsy. that CDG genes are known to cause some of those disorders. So in the big screening that people with epilepsy can get for a certain set of genes, they're now including CDG genes. So that's good because it enables everybody to say, now we have more examples of children who have disorders, and it's caused by mutation in this gene. But there's a problem. You know what the problem is? There are always all kinds of different variants. Crops up all the time. All of us are carrying thousands of different variants. And when you think about that, you say, well, how do we know if one particular one or two, how do we know if that's what's really important? Is that's what's causing what we're looking for? And if so, maybe we want to do family planning. Maybe we just want to be aware. However we do it, we need to be sure that a thing we call a variant really is a causal mutation. It really is the basis of why there are problems in this child. And why is that so important? It's not only family planning, but we might be able to do some treatments. That's why that's so important. So you got to be sure about this. And how do you do that? Well, that's where it comes back to being able to go into the research labs. Because where things stand at the moment is that many of the mutations, unless they are, how can we call them, sort of slam dunk mutations that really completely wipe out a protein, unless you have something like that, oftentimes these will become variants of uncertain significance. So in other words, the diagnostic company is saying, yeah, it could be bad, but maybe it didn't. Could be, but we're not sure. So where does that leave you? Well, that's not much help. But if that company, that hospital, that physician is able to come to research glycobiologists like us and say, we think this is a damaging mutation, but we can't prove it. Can you help us? Well, as far as I'm concerned, that's part of our job. We have to push back the frontiers of knowledge, but you know what? I mean, you're family. I mean, you are. And I think it's the obligation of scientists to do not only what benefits their own personal research, but also to do something that helps everybody else out there. If we're not doing that, what the hell are we here for? So I think that there are lots of directions we'll go in the future. One of the things I want to talk about is that you can use multiple species for trying to verify those mutations and what causes them. But not only can you do that for individual mutations, you can use genetic tricks in various kinds of systems. Fruit flies, worms, planaria, mice. You can do experiments in those that give us a much bigger picture, and we can combine all of the things we learn. Now, we may not have it all sorted out in the beginning, but we can begin to combine those things that we learned from each of those systems to kind of give us some insight into how we might be able to more accurately pinpoint what's wrong with these disorders, and maybe also a way to treat them. So I think the value to basic science is absolutely unquestionable. Their value to understanding, again, unquestionable. Their value to therapy, it's important, but it's not the end-all of the solution. Those things will provide us models where we can test potential therapies. If we think we're on the right path, we can use those models to check it. And can we translate all of this stuff that we get out of flies and mice and everything else we can find, can we translate those into some sort of cures? That's a tough go. Or at least treatments. Well, that's what's ongoing. That's why we have to keep punching the heavy bag for science to try to understand this stuff. And I think one of the things you saw come up that I think was so impressive was to see in Dr. Tadashi's work that variations in the kind of mouse, the color of the mouse, the origin of the mouse, what a difference that makes in terms of the outcome. And you're much more complicated than those mice. You saw the same thing in Dr. Chow's analysis of some of the fruit flies that he's been able to isolate, going to the market and collecting fruit flies. So that's sort of like us, coming from every place else. So we try to do things very precisely and have a single change in one mouse or a single change in one fly. And that's great. The problem is there's all this variety. And so now, even though we can have some clue, we have to start pulling this together to get an idea. Then we have to disperse it again to see if it's true in different settings. So we've got a long way to go in that. And I think if you're going to work on different models, you have to take the approach carefully. You have to think about what you're going to get out of a particular model, why you want to do it, what the advantages are, and oh, by the way, because let's say in PMM2, there are 120 mutations out of about 350 amino acids, which means every third amino acid has been mutated by nature. Which of those is the one you want to go after? And now, in what mouse background? Under what conditions in the laboratory or in the animal care facility do you want to have? All these variables are amazingly complex. It doesn't stop us. We're going to keep doing it, but realize that that's some of the complexities. And I think one of the most important things is we need to attract more interest and attention, and that's really an ongoing situation. But you know what? Advocacy. Advocacy rules. So something in the paper that said about gun control, whichever side of the issue you're on, doesn't matter. But what matters is not that politicians are saying, well, we must do this. That's coming from individual constituents standing up and saying, I'm against it, I'm for it. Whatever side you're on, it is the power that you bring in. And by being here and by doing what you're doing and showing up and being part of this whole procedure, you're making a difference. So I think that's what you need to do. Go out and make a difference. We can make a difference.
Thank you, Dr. Freeze.